So the video that you're about to view is going to talk you through the DNA Barcoding Science Night project. But first I wanted to remind you that for the Protein Database Lab Part 4 that you came up with a Science Night project. So it is possible that you will either present the Protein Database Lab Part 4 or the DNA Barcoding project as your Science Night project, depending on what data you are able to retrieve. However, you will complete both projects. Hey folks, this is an explanation of the Science Night DNA Barcoding project. So you see in the cycle beginning chart giving you an outline of the activities. And there are basically 3 pages. One would start with planning Science Night project. The engage is an article you have to read. You have to visit a website looking at previous barcoding projects, and then you engage in a discussion. This is just an overview of the process. Later, I'm going to ask you questions in a pre-lab and a proposal, and this information will be helpful for that. You are going to read another article as well as look at some previous projects that we have done, and then you're going to participate in another discussion. At this point, you have background about the DNA barcoding process, and here are some important documents. First, there's an overview document with instructions. You'll see that here gives you an outline of how to come up with your questions, timeline, and details about the actual Science Night. Then there is a spreadsheet that you're going to use throughout this process. This spreadsheet basically keeps track of all your work, all your group's work. So you can see you're filling out this spreadsheet. The orange columns are columns that I fill out. So after you submitted something and I've approved it, that's when I will say yes there. The other columns, these are things that you actually fill out. So you start off with your group members' names. You see there's information you have to fill out here, here, here. And I will change any correct or approved cells to green. So they're all red because nothing has been approved yet so far. So this will take us through the entire process. There's a proposal that you will write. This has the instructions for that proposal. And this is also one per group. Then we go on to lesson where you're going to learn more about the actual major step in the project. Once you have learned about it, then it's time to start DNA barcoding. Before you can actually start DNA barcoding, which is a long drawn out process, before you can even start, we're going to do this project exclusively in class, during class time, because I have the equipment. You're going to fill out a pre-lab, one per group, and once that's approved, then you actually get to start. And you can see here, these are the procedures, so the steps, multi-page documents. So it's going to take us multiple days to get the experiment done. Here is a document that will be helpful for the pre-lab. Recall I mentioned a few minutes ago that there's something else will be important for the pre-lab, but it's a document with information. The procedure itself will be helpful for the pre-lab. And then, all this other stuff will also be important for that pre-lab. So there's a video that talks through the DNA extraction process and more instructions for labeling tubes. And then once we send our DNA samples out, we will get DNA sequences. I will post those DNA sequences on the spreadsheet. So you can see here, another tab. You see the sample numbers. Your group will have some of these numbers, and then I will just paste your DNA sequences there. And then there are tutorials and videos that show you how to analyze those DNA sequences. We will use BLAST, but this time it will be a DNA BLAST. So here's a tutorial there. Something called a cladogram that you will be able to create using your DNA sequences. Videos here to show you how to align, find the percent identity between DNA sequences. Make sure you look at these instructions there. And then there may be some sequences that you were unable to get. This shows you how to use a DNA database to retrieve the DNA sequences that were not successful if you know the species of the organism. It's time to create your presentation, poster, etc., etc., and there are instructions there.